Please pay attention as you will be asked some questions about the content later. Information on the health risk of micronutrient deficiency and benefits of biofortified food. Vitamin A and iron are among the micronutrients that our body needs to be healthy. Anyone who does not eat a lot of vegetables and fruit, but mainly eats our common foods like gari, rice, maize, yam, etc., is likely to have very low vitamin A and iron in his or her body. People who have very low vitamin A in their bodies may not see well at night, and if they do not take care of themselves in time, can lead to permanent blindness. Also, their brain may not function very well and they may be falling sick regularly. Children who have low iron in their bodies may not grow and learn well. Adults who have low iron in their bodies get tired at all times. It may also cause pregnant women to give birth before they are due. The weight of their babies can be low, and the babies may not grow well. Scientists have found that in Nigeria, 2 out of 100 children and 8 out of 100 pregnant women cannot see well at night because of low vitamin A in their bodies. They also found out that children between 1 and 4 years are dying because of low vitamin A in their bodies. About 60 out of 100 women of reproductive age are not having sufficient iron in their bodies. For pregnant women, it is about 50 out of 100. And for children, it is about 70 out of 100 who lack sufficient iron in their bodies. To address these problems, scientists have successfully added vitamin A to cassava because a lot of us eat foods like gari regularly. These cassava varieties have been in the market and are used to make gari. Now, there are new types of cassava they have developed which will have sufficient vitamin A and iron that our body needs. Gari made from the new cassava varieties may soon be available in the market if approved by the government of Nigeria. Up till now, scientists have used 2 different methods to add micronutrients to cassava in Nigeria. These methods are referred to as conventional and genetic modification approaches. Now, they are considered an additional method referred to as gene editing. In this video, we will give you brief information about these 3 methods, while we highlight the main differences between them. Conventional method for adding vitamin A to cassava. Scientists have used this method to develop a provitamin A cassava in Nigeria. To start with, they identified 2 types of cassava. The first cassava type, which most farmers like to grow, contains little amount of vitamin A. The second cassava type is rich in vitamin A, but not usually grown by farmers. Scientists then combined these 2 types of cassava in a way that is very similar to how humans and animals meet sexually to produce their children. Thereafter, they selected the cassava varieties with the highest amount of vitamin A among different varieties produced during this process. The cassava type produced from this method has been approved by the government and is now available in the market. One limitation with this method is that only vitamin A can be added to cassava using this method. Genetic modification, transgenesis. This is a genetic engineering method used to introduce a new desired trait into the DNA of an organism by transferring the desired trait from another organism. Scientists have used this method to develop new provitamin A and iron cassava in Nigeria. They took genes that were rich in substances that can produce a lot of vitamin A and iron from bacteria and inserted them into the DNA of cassava, which was not having sufficient vitamin A and iron. The cassava type produced had been tested and grown on the field to see if it adapts to the environment and is not harmful to the body. However, scientists in Nigeria are still waiting for the approval of the government to allow farmers to be growing genetically modified cassava. Therefore, it may be released into the market if approved by the government. With this method, scientists can add more than 1 type of micronutrients, which was not possible with the conventional method. Gene editing. This will allow scientists to make certain changes to genes of the same organism without the need to transfer genes from another organism. In the case of cassava, scientists will change the sequence of the DNA of cassava in a way that will make the cassava have more vitamin A. Cassava made from this method has not been developed, but is only being considered by scientists. Scientists may also develop them if the government gives them approval. Like the genetic modification method, scientists can add more than 1 type of micronutrient to crops which is not possible with the conventional method. To summarize, the conventional method involves combining traits between one cassava and another cassava type through a natural process. The genetic modification involved transferring genes from bacteria to cassava artificially. Gene editing will involve changing the gene of cassava that is not having sufficient vitamin A and iron into a form that can make it produce sufficient vitamin A and iron.